जी sir, message. Welcome back, everyone. We have a message from Professor Dr. Ashraf H. Shalan, Chairperson ComSets, Coordinating Council. He is one of our collaborating partner for organization of this symposium. Kindly take a look.
Excellency, distinguished professors, ladies and gentlemen, good morning. Assalamu alaikum. On behalf of the Commission on Science and Technology for Sustainable Development in the South, ComSats, I extend my warm
Welcome back,
greetings to
everyone. We have a message from Professor Dr. Ashraf H. Shalan.
the International Center for Chemical and Biological Sciences, Pakistan, for holding the 15th International Symposium on Natural Products Chemistry in collaboration with ComSats, the United Nations Educational, Scientific, and Cultural Organization, UNESCO, and the Organization of Islamic Cooperation Standing Committee on Science and Technology Cooperation, COMSTEC, at this beautiful city of Karachi. It's a matter of great pleasure for me to address this august gathering. Ladies and gentlemen, natural products have become a subject of such interest and promise in the present-day research directed towards drug design and discovery. The contribution of natural products chemistry in advancement of the physical and biological sciences, its interdisciplinary domains, and development of novel applications is remarkable. The industrial utilization of plant resources, scope of phytopharmaceuticals, essential oils, food supplements, natural sources derived drugs, sustainable medicinal plants use, etc., are gaining everyday grounds. The scientific and technological progress in the era of natural products chemistry and the prominent role it played in upscaling related bioindustrial technologies strongly led to economic gains and would continue to affect the economic scenario even more deeply in the coming years. Therefore, holding of this event is timely and welcome initiative. I am certain that the event will turn out to be an excellent platform for mutually beneficial cooperation in the field of natural products chemistry. Ladies and gentlemen, ComSats member countries, including my own country, Egypt, are the lands of natural resources, where there is a valuable treasure of medicinal plants which remains to be explored. The scientific minds must come together to make best utilization of these natural capitals in order to address various global challenges. Events such as this will pave the way for sustainable utilization of these resources for facilitating socioeconomic progress. Among the most serious global issues of the recent times in the COVID-19 pandemic, which resulted in an unusual health and economic crisis worldwide, it is a matter of great urgency to reflect upon the situation and to enhance global cooperation and partnership in various areas of science and technology, including natural products chemistry, to help humanity at large. ComSats, being an intergovernmental organization, has remained devoted towards its mission of facilitating science, technology, and innovation-led sustainable development of the South, particularly in its 27 member countries. In its journey of 27 years, ComSats has made great efforts towards the capacity building of science and technology organizations in its member countries through organizing and sponsoring over 350 scientific capacity building seminars, training workshops, conferences, symposiums, and meetings in the areas most relevant to their scientific needs. In achieving its noble mission, COMSAT relies heavily on its 24 international science and technology centers of excellence, which contribute towards organization's technical program through pooling of human and technological resources. These include ICCBS, which has been playing an important role in achieving the objectives of COMSAT's network through organizing various capacity building activities, training scientists and researchers, and taking part in collaborative R&D initiatives. I would like to once again thank ICCBS for collaborating with COMSAT, UNESCO, and COMSTEC to organize this symposium. I would also like to acknowledge the facilitative role played by the Pakistan National Commission for UNESCO, PNCU, in this regard. I thank the eminent subject experts who are taking part in this event for sharing this experience and knowledge in this vital field. I'm sure that the event will be able to meet its objectives and will act as a forum for advancing R&D collaborations in natural products sciences. Thank you so much.
With this, we are going to start our first technical session, and to chair the session, I invite Professor Dr. Auditorium.
Good morning, everybody. I would like to welcome you to the introductory plenary lecture in this symposium. And I would like to welcome Professor Ataur Rahman for this very first lecture. You know, this lecture is very well deserved, and I will give a short excerpt from his scientific and societal achievements. Professor Ataur Rahman made his PhD in organic chemistry from the King's College, University of Cambridge in 1968. He has published over 700, precisely 781 research publications, 50 international patents, 70 chapters and books, and 366 books published largely with major and European presses. He's also an editor of a journal. Besides these very prestigious scientific achievements, Professor Ataur Rahman has served as a Federal Minister of Science in Pakistan from 2000 to 2002. He was a chairman of the Higher Education Commission. He has been elected as president of the Pakistan Academy of Sciences twice, and he is currently president of the Network of Academies of Sciences of Islamic countries and foreign fellows of the Chinese Chemical Society. Actually, two institutes hold his name at several different universities in Malaysia and in China. And you have already heard about all of his prestigious prizes that he was awarded to. And I will not repeat them here, but I would like to welcome Professor Ataur Rahman to his lecture.
Thank you, Madam Chairman. It's a pleasure to be here. And I'll be talking to you today a little about natural pot chemistry, one aspect of natural pot chemistry. And then I will go on to talk a little bit about higher education and what I try to do in science and technology in the country. And then later, I will also talk about the future, what areas we are working on, and from the Pakistan scenario, in terms of science and technology. So it'll be a bit of a potpourri where I'll be talking about different aspects of some of the things that we have done. Natural pot chemistry continues to be a source of great wealth because of the wide diversity of structures and the corresponding diversity of the geographic regions from which the plants are isolated. And one big challenge for developing countries like Pakistan is the single compound, single active compound syndrome, I call it, because without having that, you cannot get FDA approval. And in order to go through the process, it literally costs billions of dollars of highly specialized companies, international companies, for which the facilities just do not exist in our part of the world. But there is an exciting transition which is taking place right now. The awareness that if a compound mixture is active, and if it is safe, if it is backed by appropriate clinical trial data, proper double-blind clinical trials, and if the key components which are responsible for its bioactivity, irrespective of their structures, have been identified in the form of biomarkers, specific compounds that are necessary, then there's no reason why that whole concoction may not be introduced into modern medicine. And that is indeed the way forward. And one recent example of that is against COVID, where we undertook in our institute clinical trials of a herbal preparation, Chinese herbal preparation, which was found to be very effective, as good as the best antiviral medicines, remdesivir and others available in the West. And it substantially reduced the period of recovery of COVID patients in those double-blind clinical trials from an average of 10 days to 6 days. And it was found to be extremely effective. And this is now already used in China for treating COVID patients, and will now be introduced also in Pakistan. And hopefully then it will be followed by in other countries. So as long as you have solid clinical data with respect to efficacy and safety, backed by reproducible clinical trial data, then there is no reason why we should be trapped in the single compound syndrome. When you are eating an apple, you are taking thousands of compounds in you, and you don't worry what the structures of those compounds are and what the activities are. And the same should apply to herbal medicines, provided they are backed by proper scientific data. So this was my first comment, the growing importance of natural materials for the treatment of diseases, provided that we adopt and adapt this new scenario and implement it. And there is much that we can learn from China in this respect, who have a highly documented system of traditional Chinese medicine, TCM. And more recently, in the last about 5 years ago, China said that we will abandon clinical trials on human beings. These are laws that they've passed within China for herbal extracts, for TCM. As long as the data on animals is effective and satisfactory, then China has now allowed the introduction of herbal extracts and herbal materials and natural materials directly into modern medicine in an integrated approach to modern medicine, where herbal drugs and modern compounds, synthetic compounds, go side by side and are administered side by side. Well, having said that, this is a joint presentation which I'm making with my dear friend Iqbal on many of the compounds that we have worked together Pakistan, you find amazing geodiversity from the deserts of Sindh to the fertile plains of the Punjab to the towering mountains of the Himalayas. You have an amazing diversity, geographical diversity. And this is accompanied by a corresponding structural diversity in the plant kingdom. So these are sources of some very exciting and interesting compounds. And over the years, we have been looking at that. This institute started as a purely chemistry institute, but now it's expanded into many areas of biology, which I indicated this morning, because biology is nothing but chemistry at the molecular level. Ultimately, it's all chemistry. With my due apologies to any biologists sitting around here. So it's all chemistry. And so it's now, you find some very interesting experiments being done here in various biological areas. So this is the biodiversity that I was talking about, from the south of Pakistan, the desert areas, to the northern Himalayas, 8 climatic zones, and the diversity of climates and compounds. And as Dr. Iqbal mentioned, we have over the years worked on some 2,200 plants. We have extracts of them also available, some 6,000 extracts. And this is one of the largest collection of extracts and pure compounds that we have, a bank that we established in our institute. And in my own work, we have isolated about 2,200 new compounds, novel chemical entities, varying structures from indole alkaloids, isoquinoline alkaloids, triterpenoids, terpenes, various types of terpenes, flavonoids, and some 83 students have completed their doctorate degrees under my supervision. And I'm proud to say that Professor Iqbal is one of them. So it's a real pleasure, Iqbal, to have bright stars around us. And there are many others, Dr. Zahir, and there are many others sitting around who are all my students. So here I would like to talk a little bit about perhaps the next frontier in science, the new horizon, and that is our own human brain. Amazing, amazing structures. The ability to learn, the ability to store knowledge, the ability to recall, the basis of emotions. 100 billion neurons, almost 100 billion neurons, almost the same number as the stars in our galaxy, 100 billion. And each neuron talking to some 8 or 10,000 other neurons. So arguably the most complex object of our universe is our own brain. And understanding the chemistry of thought storage and thought recall is one of the most exciting areas of science that I find today. Fascinating area. And it's an area that we know very little about. The neurodegenerative diseases such as Alzheimer's, Parkinson's, epilepsy, autism, etc. What goes wrong at the molecular level, we still are struggling to understand. There is some knowledge, but what is going on at the molecular level, until we really understand the functioning of the human brain, we will not really get to grips with the basis of those neurodegenerative diseases. A lot of understanding has come through the work over the last 3 decades of people like Eric Kendall, the Nobel laureate, and many others. But these are at a different level. It's about synaptic plasticity, about how neurons interact with each other, about long-term memory storage and about short-term memory storage, about the involvement of genomics in this area and the genes responsible for such storage systems. But actual type of storage that is taking place and how, what is the basis for the knowledge storage and how the various facts that are stored are able to interact together so that the process of logic can then develop, is a fascinating holy grail of science today. So I'll be talking a little bit about epilepsy, some wonderful work done by Dr. Farzana here. She's with us, and her colleagues, and Dr. Iqbal. And this work is being carried on and we are now not only into epilepsy, but also looking into Alzheimer's, into Parkinson's, and so on. And Farzana and Iqbal perhaps will talk some of the work, but I'm just giving you a little bit of an overview of some of the wonderful efforts that are being made in the area of understanding the functioning of the human brain. So epilepsy is a neurological disorder which affects about 5 to 60 billion people, and it can have many causes. They can be genetic, congenital, they can be caused by head injuries or by ischemic injuries, and there is no cure. The medicines today are as good or as bad as they were some 200 years ago. So, and the reason that there is no cure is because we don't understand the neurological processes at the molecular level in sufficient depth to understand what exactly is going wrong and what kind of mechanisms, disease mechanisms, do we need to intervene in order to develop a cure. So, looking at herbal drugs and looking at possibilities of finding herbal extracts and then pure compounds, which will be effective for such neurological diseases as epilepsy and for other neurological disorders, is one of the goals that we have strived for. And the first line of treatment are sodium channel blockers, and there are only a limited number of anti-epileptic drugs which are available that can suppress seizures. So when you are having a seizure, when you are having an epileptic fit, your individual neurons, they go through an uncontrolled, sustained, repetitive firing, these neurons. And this is what causes, appears as epilepsy. So there are various mechanisms to try to control them. So we started looking at various medicinal plants. Nationwide ethnobotanic survey was done, and some plants which appeared to be useful and had been claimed to have useful properties for epilepsy included Delphinium denudatum, Aconitum species. There are 3 different Aconitum species which are mentioned here. And so what the epileptic patients do, this gentleman is not an epileptic patient, he's just wearing a necklace, he's a laboratory attendant in our laboratory. But the tubers of this plant, Delphinium denudatum, are worn like a necklace around the neck, which indicated to us that the compounds might be volatile and might be in small molecular weight. And there are various bioassays which are used for looking at new anti-epileptic drugs. You can have electrical and chemical tests. You can have a sustained repetitive firing bioassay induced in neural cell culture. And then various anti-convulsant drugs are available, such as phenytoin and others, to look at the suppression of these of this neuronal firing. And in the Panjwani Center for Molecular Medicine, just across our buildings here, you have the patch clamp facility where you can artificially induce repetitive firing, simulate epilepsy by passing very weak currents of about 10 to the minus 12th of an ampere. And this results in uncontrolled repetitive firing. And then you can expose those neurons to plant extracts or to purified compounds and see to what extent these are suppressed. And using this facility, patch clamp facility, what we are actually looking at is the very weak currents caused by the flow of sodium, potassium, calcium, and chloride ions. So using this technique, we can monitor if an extract is active, if the purified compounds are active, and then try to identify the compounds through a biodirected fractionation assay. And using this technique, we isolated a whole series of compounds. They were small molecular weight isoxylatones. They are just drawn in various colors to indicate their biological activity. And so the two compounds, the E and Z isomers at the double bond, the double bond at the one position, these two isomers are found to be very, very active. And we then, both of these were found to reduce the expression of the C-phos protein, which belongs to the IEG, the immediate early gene transcription factors. And expression of this C-phos protein is known to increase during epilepsy. So these were found to suppress this, and they also accepted, and therefore was shown to have potent anti-epileptic activity in mice models. And the neuronal activation was studied through immunostaining techniques. Also, the brain-derived neurotrophic factor is again a protein of the neurotrophin family of growth factors, and its levels are known to increase in epilepsy. So this again was found to be suppressed through the use of isoxylatones in kindled animals. So something exciting was discovered here, and the fact that they're simple means that we can synthesize them also in kilogram levels, and so it's very easily accessible. And whole libraries of these compounds were synthesized, starting from just isophorone, which is commercially available, through Horner-Wadsworth-Emmons reaction, functionalization of the ketone group, followed by hydrolysis, and then reaction, preparation of the binder amide and Grignard reactions to synthesize various types of isoxylatones functionalized. And there were a whole range of isoxylatones synthesized in this manner. And this was internationally patented. And so these compounds have gone through phase 1 clinical trials and have been also into phase 2 clinical trials, and are in the process of development. This whole area led us to be thinking about memory in general and the processes through which memories could be stored in the human brain. And there are many reviews and articles, etc. Those of you who are interested, I'd be happy to share with them. But they make fascinating reading, understanding the basis of human memory storage. And so what we proposed some years ago was that the hypothalamus of the human brain contains glycoproteins. And these glycoproteins, there's a peptide backbone with various sugars, glucose, arabinose, fructose, xylose stuck onto them. And these are bristling with hydroxyl groups. So nature has formed a large number of, a kind of a velvet, in which the strands of a velvet, the kapra dikhte hain, velvet ka, usme dhage jo hote hain. So it's like these hydroxyl groups are like this velvet. And through hydrogen bonding, you can have the freezing of the conformational mobility. Normally they're waving about. But as you look at an object, the conformational mobility can be frozen through hydrogen bonding. The greater the number of hydrogen bonds, the greater the strength of the memory, a theory. And so, and the breaking, so learning then can be regarded as formation of patterns through hydrogen bonding in this natural velvet of hydroxyl groups, intramolecular, intermolecular hydrogen bonds. And Dr. Zahir has also looked at some of the computational models that can be looked at to see the energetics that we have that may be involved. And so formation of hydrogen bonds, the freezing of conformation mobility and pattern formation as a result can be the basis of learning, and the breaking of these hydrogen bonds can be the basis of forgetting. And the interrelationship of these patterns can be considered as the basic of logic. So emotions and interrelationships. So these kind of intermolecular or intramolecular hydrogen bonds between hydroxyl groups of sugars with other sugars or with nucleic acids present can be the basis of pattern storage. And there is growing evidence that this might be true. There are many articles which have appeared which seem to back this hypothesis, but still it remains a hypothesis because functional MRI has not grown to a point where you can actually look at the molecules themselves. But time is going to come whether we'll be able to tell whether that indeed is correct or not. So this is a very exciting area of research, understanding of neurodegenerative diseases, understanding the human brain and looking at natural products which can be used for treatment of these neurodegenerative diseases. Let me now switch gears and talk a little bit about some of the work in the higher education area, in science and technology area, developing a knowledge economy. I was the Minister for Science and later the Chairman of the Higher Education Commission. And in those early days, that was between 2000 and 2008, we managed to get substantial funding for science and for higher education, some 6,000% increase in the development budget of science, and about 3,500% increase in the development budget of higher education. And we focused on quality, access, and relevance, the three major parameters of education at the higher education level. And basically, our focus was on faculty development. Universities are not about beautiful buildings, about instrumentation, about equipment, all that happens. The first and most important thing is high quality faculty, bright young men and women who are able to discover new things, who have energy, who have passion, who are willing to work hard day and night. And so we sent about 11,000 students abroad to world's top universities after very, very intensive selection processes. And each one was supported with substantial grants. Now it costs about 3 to 4 crores of rupees to train one person at a PhD level in a good university abroad. But sending them abroad is only part of the game. We must create the ecosystem that they come back and they contribute to the development of your own country. That's perhaps the even more challenging part. Finding scholarships and training them abroad is you are spending your nation's money to help other countries. If they settle in USA or in Europe, then it's our money being spent to develop other countries. We shouldn't be doing that. We should be investing our money to develop our own countries. And so we did basically 5 things, 5 or 6 key things. There are many other measures. One, we dramatically changed the salary structure. So the salary of a professor became 4 to 5 times the salary of a federal minister in the government under the new tenure track system. A contract in which you are evaluated 6 times internationally. Twice as an assistant professor, and then you win tenure. Twice as an associate professor, and then you win tenure. And twice as a full professor. So it's a contractual system with regular international evaluation. But a completely, so if you're good, and if you think you can make it, then you join this system, and then you go up the ladder. You of course get much more salary than others. So this was a contractual system, and this still exists. Over the last 2 decades, it had gone down, but I'm pleased to say that over the last 6 months that I'm involved again in the higher education area as the chairman of the Prime Minister's National Task Force on Science and Technology. There's been a decision a few months ago, which the Prime Minister took, to give a substantial increase, 35% increase to everybody, and 100% increase to the best of the tenure track salaries. So there's going to be a substantial jump. So one step was having a good salary structure to come back to. The second was research funding. When they come back to a weak university, there's no money. What do? So we said every student who's coming back can apply for a $100,000 research grant 1 year before their return. Of course, all projects will go through an extensive peer review, but so that even if they come back to a weaker university, there will be a $100,000 research grant, up to a $100,000 research grant available. So access to good research funding. Third was access to literature. A digital library was created for books and journals, so 65,000 textbooks and 25,000 international journals through the digital library. One important hurdle for researchers who come back, young people, is access to sophisticated instrumentation. Not every institute in the country can have these kinds of NMR or mass spectrometers that we have, for instance, in this center. We have about 13 superconducting NMR spectrometers, including an 800 megahertz. And we have about more than 20 mass spectrometers of different types. So we brought in a system that every researcher will have free access to scientific instruments. And the Higher Education Commission will pay for the analysis. So that so they can send their samples to any laboratory of their choice, whether it's PCSIR or NASCOM or KRL or this center or wherever, and the analysis will be done within 72 hours free of charge. So the excuse that we cannot do this because we don't have that evaporated. And so you have free access. So that again, system had in the last few years had gone down, but that is again being revived now. And of course, jobs on arrival. That is because one of the most frustrating times that a young researcher has is when he or she comes back with a PhD, goes around hunting for jobs. Universities take about a year to advertise and so on. The selection boards, of course, they have a certain criteria. So we had a matchmaking process which started more than a year before they were returning. So that and once they landed for the first year, the jobs will be guaranteed by the HEC. They will work as HEC scholars, and subsequently they'll be absorbed into the relevant universities with additional funding going to the university, so that universities will have additional PhD level faculty. And centers of centralized instrumentation centers were set up in many universities, and many other measures were taken. And these and others led to a 97.5% return rate. So for every 100 scholars going abroad, 97.5% were coming back and joining Pakistani institutions. And that resulted in a remarkable change. And I think there is no other country in the world, correct me if I'm wrong, which can show this kind of a development. This is Pakistan versus India. Year 2000 to 2020, a 20-year period. Number of publications per capita, per 10 million individuals. So year 2000, Pakistan was 44 publications per 10 million population. India was 172. So we were 400% behind India in the year 2000. And then our policies came in, and our young people were sent abroad, and they came in, and research was. We also had very liberal research funding available through the HEC, and the NRPU program, the National Research Program for Universities, was one of the most dynamic programs for supporting young scientists, and through which you get substantial. Now that is again being revived now by me, with 20 million rupees grant going to young scientists under the up to 20 million under the NRPU program. And you see the green, Pakistan. India is red. So Pakistan going up and catching up and exceeding India according to the Web of Science data in the years 18, 19, and 20. And if you look at the Scopus data, the graph is very similar. We are about, this again is this data from Scopus, and it's about the same as India. So something quite remarkable has happened in Pakistan. And there have been many articles written about it internationally by the chairman of the UN Commission on Science, Technology and Development, and many others. Thomson Reuters came out with a report, another brick on the wall, BRIC, comparing Pakistan with Brazil, Russia, India, and China, and the number of highly cited papers from Pakistan. The percentage, not the number, the percentage of highly cited papers was actually much better from Pakistan than any of these countries. So, so there have been internationally neutral international praises for what was going on. India became concerned and a presentation was made to the Indian Prime Minister. And India then decided to abolish its University Grants Commission and set up the HECI, the Higher Education Commission of India. So a lot of other things were done, including I was also the minister responsible for IT and telecom, so promotion of IT, promotion of mobile communications, etc. I won't talk about that because I would like to move on to what is going on now. Something quite exciting has happened in Pakistan in the last 2 decades. And I'm glad that I had a small part in this. More recently, now let's turn to Prime Minister Imran Khan and what is happening now. So I met Imran Khan about before he became the Prime Minister. And I told him that Pakistan is a very lucky country in many ways. Perhaps the most important wealth that we have is our children. 67% of our population, 67% of our population is below the age of 30. So we have a very large young population. It's a huge demographic advantage. So I said that if you want to develop this country, you have to invest in education and not just higher education. It has to start at the bottom. So primary, secondary, technical, and higher education. We have examples of Sri Lanka where you have excellent low-level education, but higher education will remain weak. There are other countries which have developed higher education. It has to be a strong pyramid. The base has to be strong, and so has to be the tip of the pyramid. So it has to be all the way, and then it has to be connected towards socio-economic development. So transitioning to a knowledge economy, that's extremely important through investments in education, in science, in technology, and innovation. So a number of task forces were formed by the Prime Minister, and I'm chairing the task force on science and technology. I'm also co-chairing the task force on information technology and telecommunications. But most importantly, there is a task force on knowledge economy, which the Prime Minister himself chairs, and I happen to be the Vice Chairman. And 6 federal ministers are members of this task force, which include our federal ministers of finance, planning, education, science and technology, IT and telecom, etc. And under this, about 100 billion rupees worth of projects are being implemented today. That's really a huge amount when you consider that the total budget of the Ministry of Science and Technology was only 1 billion 3 years ago. Now we have about 100 billion rupees worth of projects being implemented. And Pakistan is moving forward very rapidly towards developing a strong technology-driven knowledge economy. And so the key is the ability for countries to manufacture and export high technology goods. That's where the money lies. The ability of countries to manufacture and export high technology goods, whether they are biotech products, pharmaceuticals, engineering goods, IT, and other high value agriculture, etc. So there is, I'm setting up a number of new universities, and a lot of my time is now undertaken by these efforts. One new university which has started functioning is near Islamabad. If you go to Islamabad, if you have a little time, I'll be delighted to encourage your visit to the Pakistan Austrian University of Applied Science and Engineering in about 45-50 minutes drive. And this is the first university in the world, to my knowledge, where the students will get degrees from different foreign universities, depending on which department they are studying. So each department is being godfathered by one foreign university. This is my idea, which, you see, if you are collaborating with a foreign university, it's very difficult for them to set up an entirely new, or help to set up an entirely new university in a far-off region like Pakistan. But if you ask them, can you help us develop one department? It's much easier. So we have 8 foreign universities now involved in the Pakistan-Austrian University. There are 3 from Austria and 5 from China. And depending on which university you're in, you will get double degrees or joint degrees with your university as well with the foreign partner university. And there are 3 streams: a Fachhochschule stream, which is a high-level technical training at bachelor's and master's level, a graduate research stream, and then a technopark. These are the 3 streams on which there is focus. So this is the new university. This was inaugurated last year. It took only about 2.5 years to have the whole thing in place, and it's now operational in Haripur. And I'm now setting up similar universities, and these are the various partner universities in China and in Austria, which are partnering in this development. And now we are working for another university in Sialkot and a 3rd university in Islamabad. These are now, so the winds of change are now blowing, and these are being funded by the provinces and by the federal government. And as a result of this, a lot of knowledge economy. We also have programs at the school level, metric tech, so that students at the metric level get technical education. That is also has now started, and also there are other projects underway. So I would just like to stop here and say that the real wealth of a country lies in its children. We have to invest in education, science, technology, and innovation and entrepreneurship. And in order for that, the vision and the strategy has to come from the top. And that's what I'm trying to do, trying to convince our leaders that the way forward for Pakistan is not just building roads and bridges. It is building highly creative young men and women who can contribute to society. So that the real wealth of Pakistan, dear children, is you.
Questions to Professor Adam?
Yes, Iqbal.
Thank you very much, sir. Always inspirational from the spirit. I just wanted to know, I mean, with these universities, lots of graduates will come in with high quality trainings. What plan we have for them to be productively engaged in technology development and others? So what is the next part of the ecosystem?
Yes, that's a very important area. In fact, the heart of these universities are the technoparks. And these new universities, what we have said is that your success will not be measured just by the H-index or citations or impact factors or patents, but the actual commercial products that are produced from your university. So we have set aside, for instance, in the Haripur University, about $10 million just for new product development. And we have Austrian and Chinese industries on the one hand, and Pakistani industries on the other, focusing on specific products that we would like to develop together. So, for instance, one of the areas for the Haripur University, the university in Haripur, is that of minerals. Pakistan has huge mineral resources, and these are just dirt cheap. They are just sent out. So we have the entire technology of extraction, purification, and export of minerals is going to be done here in this university. Similarly, there are a number of products that have been identified. At the moment, there are 10 commercial products. There are actually 30, but there are 10 commercial products which are well on their way to development. So the linkages between the university with industry and agriculture are critically important. And so that is the primary focus, and that is how these universities will be different from the general universities that exist in Pakistan.
Okay, Dr. Arman, thank you for that wonderful presentation. You showed those very impressive graphs where there was a substantial increase in the number of publications from Pakistani universities. And that's very good. But don't you think there should be a greater emphasis on the quality of those publications rather than the numbers? And what are
Absolutely.
you trying to, what are you doing about that?
Yes. Firstly, these are all publications in impact factor journals. So all these publications, those which are not in impact factor journals, are not counted. So that's the first check that we have. Second is that one looks at the number of retracted papers from Pakistan. And there were some news items about it some time ago. And I looked at the data. These are about 0.1% of the total number of papers being published. And then I compared the data with India and other countries, and it was compatible. So about 1 in 1,000 papers are being retracted. So that is there. Third is the highly competitive environment within the universities themselves that we've tried to create. That in order to go up the ladder, you will be looked at from the point of view of your citations, your H-index, the impact factor of the journals, the patents, and also your resourcefulness, your ability to win funding, and your contributions to society. So we, for instance, in this institute have a very clear matrix, which Iqbal could share with you, about how we evaluate our faculty. And we are encouraging our other universities to adopt similar evaluation criteria for faculty members, so that the quality, the competitive quality becomes integrated into the system of their operations. It has to be, the government has to provide facilitation in terms of equipment, infrastructure, facilities. So it has to be a two-way street. It was happening back when Musharraf was there, but then the support kind of waned off. And so the momentum was, to a great extent, stopped. But now it's beginning to happen again. And that's what I said for the future, to look at the new horizons and then to invest in them and see the country change.
We can take one more question, and then I would like to ask you to keep your questions for the lunch break, where you can ask Professor, because we have to proceed on the next section.
Thank you very much, Professor Akram, for a very nice lecture. I have one question. Nowadays, in our country, Iran, people talk about the third generation university and the involvement of professors, university professors in science-based company, making science-based company. I wanted to know what's your advice and what's your idea to do so by making a company by university professors, because research and maybe commercial aspects of research are very different worlds. So I wanted to know what's your advice. Thank you very much.
Yes, well, that's a very important thing. In Pakistan, unfortunately, it was forbidden by the Higher Education Commission for university professors to work in industry. So I then talked to the Prime Minister and I said, this is absolute nonsense. We must have our professors having the freedom to work in industries, and a part of the time they must work in industries. And we can learn from China, from Japan, from Iran, and many other countries, how this has been a deliberate policy for many countries to encourage our professors to work in industry. So this has now changed, and the notification has already gone out from the Higher Education Commission a few months ago to all vice chancellors, encouraging them to work in industries, and that this would be one area that we counted towards their promotion, etc. So that kind of a linkage is extremely important. And I think what Iran has done is absolutely fabulous. And I think what has happened in Iran is because of the sanctions. Perhaps you would not have evolved so quickly, but that had the sanctions not been in place. So I think you should send a thank you note to the United States saying thank you for putting sanctions on us, because you have allowed us to evolve so quickly as a result. But I think what you have done in Iran is wonderful, and that's also something that we would like to follow, as has been done also in China and other countries.
Maybe as a last comment, I would like to say that we must also have a very efficient and high quality basic grammar school and high school system, because otherwise we could not proceed maybe with this very extensive and impressive extension of the university system.
Yes, and that is extremely important. Unfortunately, the lower level education is fragmented, and there's much that we need to do to improve the lower level education. So we have some very good schools, but we have a large number of very weak schools under the government system. And that's now an area that I'm working on to change the foundations, strengthen the foundations to encourage the development of creativity.
And then I would like to give you a present for your presentation and for your engagement. Now we come to the next speaker, who is Professor Anna Shubanisam from the University of East Anglia in the United Kingdom. The title of his presentation will be Gold from Old, Awakening Silent Biosynthetic Tree Clusters. Professor Ganeshan is a committee member of the Drug Discovery and Development at the ICBs, and he is also a committee member of the Bioorganic and Medical Chemistry sector in the Royal Chemical Society, and a co-founder of a company, Carlos Terpoy. Welcome, and we're looking forward to your lecture.
Thank you for the kind introduction. And firstly, I'm grateful to the organizers for inviting me. I visited the Institute, I think 13 years ago, and it's a pleasure to be back and see all the changes and improvements. It is, of course, a world-renowned center for natural products, so it's great to be here. Not everyone might know, but I'm from Malaysia, and I'm named after a Hindu god, in case you did not know that. And I actually live in the UK. This part of the campus where I work at, which is the University of East Anglia. And I didn't put in an introductory slide, but what's not on in the presentation is that, in fact, in the UK, where I work in Norwich, is a major center for natural product research. And there are many colleagues of mine who work on natural products in all aspects, ranging from the more basic science to commercial food products and agrochemicals. I'm summarizing here humans' interaction with natural products in a few key moments. And since prehistoric times, for thousands of years, we have exploited plants, of course. And more recently, in the 20th century, the microbial world as well. And this, in fact, vancomycin, the producing organism, came from Malaysia. So it's a nice, although exploited, of course, by the Western pharmaceutical industry. And since then, we've also moved to more unusual ecological habitats, including marine organisms. And what we're seeing in this century is the tremendous advances in technology and data processing and computing that allows us to carry out experiments that would not have been possible even 20 years ago. And there's just one example, this from the Dorstein group in the US. And this is based on taking hundreds of samples from all over the human body to map the distribution of 3 different bacterial genera And one of the subjects is a female and the other is a male. And you can see the distribution of bacteria in our bodies is quite different based on location, but also on gender. And this is something that would have been impossible to do not so long ago. And in my group, we work on traditional plants as well. But for today, I'm going to focus purely on some microbial results, which are part of one student's PhD project. And recently, the microbial natural product world is an area of really exciting research. And that's because in microorganisms, the genes that are involved in natural product biosynthesis are usually found next to one another in what we call a biosynthetic gene cluster. And these gene clusters, we can now readily identify because they do predictable chemistry. So for example, the fatty acid synthase in your bodies carries out a Claisen condensation. And to some extent, that enzyme will resemble the Claisen condensation enzyme involved in, say, tetracycline biosynthesis in bacteria or in tetrahydrocannabinoid synthesis in plants. So by looking at the architecture of these enzymes, you can assign function to them. And we don't even need to do that. You can do this in an automated way using computer programs. So you can identify what enzymes are carrying out and and you can then interrogate sequence genomes, of which there are thousands now available publicly, and assign putative biosynthetic gene clusters in these sequenced microbial genomes. And what we find by doing that is that in reality, the natural products that you can identify from a microorganism are much, much smaller than the total number of natural products than the total number of biosynthetic gene clusters present. And this is simply because when we culture these microorganisms in the laboratory, these are very artificial conditions that are very different from their normal habitat. We give them plenty of food, there's no competition. So usually they do not feel the need to be turning on secondary metabolite production. So these genes are silent. And the question then arises, can you activate these genes? And if you can then produce, induce the species to produce lots of wonderful new compounds, even from very well-known microorganisms that have been studied for many decades. And there are different ways you can do that, which I've summarized here. One is simply by manipulating the growth conditions of the microorganism in the laboratory. You can also use what we call small molecules that are added to the fermentation. And we'll see one example later. And you can also do more modern technologies such as genetic expression or deletion of certain genes to see if that then changes the biosynthetic production. And last but not least, if you know what the biosynthetic gene cluster is and you have difficulty expressing it in the producing organism, this can then be cloned and reconstituted in an artificial host. So you're even bypassing the producing organism in nature. And there's a nice recent review which gives you some examples of the types of experiments you can do. So you can change fermentation conditions. You can also use epigenetic modulators, and you'll see an example later in the talk. These are more molecular biology-driven approaches where you can clone in or delete genes. And all these different approaches have their pros and cons. The simplest is the one that involves changing your fermentation parameters. However, there are many of them, and you can literally, with one strain, carry out thousands of experiments to see if there's a change in metabolite production. Where some of these are more sophisticated and pathway specific, but they then require more molecular biology techniques and gene cloning or gene editing. And here you see some of the ways in which you can use this information of gene clusters. For example, if you know the structure of the natural product and you know the biosynthetic gene cluster, then you can do a search of the database to see if another organism has a similar biosynthetic gene cluster, and then you can go after that other natural product. Meanwhile, you might also be in a situation where you know the structure of the natural product, but you do not know the biosynthetic gene cluster. But in that case, you can use your knowledge of biosynthetic pathways to predict what the BGC should look like. And this one example here, if you look at the structure of Griseofulvin here, looking at the structure, there's 3 methyl groups added. There's a chlorination here. There's a double bond that's being reduced. So the biosynthetic gene cluster should contain enzymes for all these functions. And you can then go through the sequence of the genome to see if we can predict a likely candidate biosynthetic gene cluster. And also, if you don't know the structure or the BGC, you can compare genomes from different organisms and then use that to try and identify gene clusters that are homologous to each other, which suggests they're producing natural products that are similar in structure, and try to identify them as well. And excuse me, yeah, I'm summarizing the epigenetic approach. And this is the DNA, the way it looks in cells. There's the double helix of DNA, which is wrapped around histone proteins. And both the DNA and the proteins undergo structural modifications, which are reversible. And the 2 major modifications are, firstly, for the DNA double helix, it's methylation of cytosine residues. So there are enzymes that methylate cytosine to make methyl cytosine, and also enzymes that then remove it by stepwise oxidation of the hydroxyl group. Meanwhile, for the histone proteins, one of the major modifications is the acylation of lysine residues. This is carried out by acetyl and acyl transferases to put acyl groups on the lysine residue. And these can then be removed by hydrolyzing enzymes. And at the moment, there are over 50 small molecule drugs in clinical development that target these epigenetic pathways in various stages of clinical trials. And there are also some approved drugs, 2 of which are shown here. This is azacitidine. This is an inhibitor of this step here, the DNA methylation step. This is vorinostat. This is an inhibitor of histone deacetylases, which is this step here, the hydrolysis of acetyllysine. And these are drugs that are approved for oncology, and they work by basically activating genes. So the question is, if they activate genes in humans, can they also activate genes in microorganisms? And the answer is yes. So you can add these compounds to a fermentation culture, and hopefully they will then induce biosynthetic gene cluster activation. Not always, but sometimes. And everything I'm going to show after this is the work of this PhD student, Muhammad Al-Dolmi from Saudi Arabia, who since now graduated and is now a faculty member in the Imam Abdul Rahman bin Faisal University in Dammam, Saudi Arabia. A really enthusiastic student. He came from a more traditional pharmacognosy plant background, but he was willing to listen to my ideas and teach himself how to do fermentations. And once you can do fermentations, it's actually not difficult to do natural product chemistry with microorganisms. And this is one of the projects he did. And here we are looking at Aspergillus fungi, and we deliberately chose 5 well-known Aspergillus species for which the complete genome sequence is already available. And we deliberately chose species by which, through bioinformatics, we can show that there are many putative biosynthetic gene clusters present. So all of these 5 species, within them contain 50 to 100 putative biosynthetic gene clusters. At the same time, if you look in the literature on those 5 species, there are only about 5 to 10 natural products that have been reported from those species. So potentially, they are capable of producing many other natural products which have not been observed in the laboratory. And the experiment itself is fairly simple in the first stages. So these are quite small scale fermentations. And we do 3 fermentations per species. One is simply a control. And in the second and third, we are adding these drugs, either azacitidine or verinostat. We also do the experiments with a combination of the two to see if they are synergistic. And then we do, the hard part is then comparing the metabolomics of these different fermentations. So this is where it becomes challenging. This is a very small set of experiments. You can do literally hundreds or thousands of these, but then you need to process each of these samples and compare their metabolic profile against each other. And over here, you see the HPLC traces. This is the control with this species. And we can see that when we add the drug verinostat, it actually shuts down a lot of secondary metabolism. You see a much simpler HPLC trace, and here's one major induced peak. And over here, there's another one. It's a different Aspergillus species, not so interesting in terms of secondary metabolism in the control. And you see one major induced peak over here. You also see residual amount of the drug itself, verinostat. So out of these different fermentations, we saw induction in 2, which is roughly like a 20% success rate. I think Mohammed was disappointed at this, but I would say this is actually quite high. So normally when you do these types of experiments, you have to do lots of them with many species in order to get some inductions. Now, what you see in the HPLC is only the scratching the surface because it's limited by the sensitivity of your instrument. And nowadays, the natural product chemist's best friend is the mass spectrometer, because you can interrogate samples at a much higher level of resolution and sensitivity. And what we did then is look at these cultures in an LCMS mode, and you'll see a much more complex picture here. And I'll need to walk you through this. So this is basically based on 6 replicates, and we do these LCMS analysis. So each of these dots you see here is a separate feature in the mass spectrum. So compared to the much simpler HPLC, we are now seeing, if you like, the total metabolome of this species, and you're seeing literally hundreds of components in there. And the reason it's so complex is because mass spectrometry is incredibly sensitive. So you would not be able to isolate any of these compounds in a practical manner from that fermentation because they're produced in such small quantities, but you can detect them. And the way we analyze these things is by these so-called volcano plots. So I'll tell you how to interpret these. It's a nice way to compare side-by-side data for experiments often used in genomics and proteomics as well. And what we're doing here on the left quadrant here is the control fermentation, and on the right is the fermentation with the drug verinostat. And on your x-axis is the fold difference in expression between control plus drug. So the more you go to this right-hand side, the more there's an increase in the expression with the drug compared to the control. And in fact, you see there's a lot more dots on this side, which is what is also reflected in the HPLC. So when you add the drug, the metabolism is mainly shutting down. So you see more metabolites produced without drug, but nevertheless, there are also some metabolites induced as well. All these dots over here. That's on the x-axis. And then the y-axis is a statistical parameter. So the higher up you go in the y-axis, the more statistically significant these differences are. The real question, of course, is what all these hundreds of molecules are. And in fact, we don't know. So we had a list of metabolites that should be produced by this species. We also had lists of natural products produced by other Aspergillus that are closely related. And in fact, with all that information, the only metabolites that we could identify for this species are these 3 dots here. So we still do not know what these other natural products are. And for the other fermentation, we could identify too. So that tells you how complex secondary metabolism is, even in a very well-known species. And I'll tell you a bit more about how we identify these compounds here. And this is one of them. So this is the amidacelamide, which is two related lipopeptides. And they have, in the literature, characteristic fragmentation pattern in the mass spectrometry. And we identify all these fragments in our sample. So we are confident that it is the same natural product. Furthermore, when we do bioinformatics, and I'm not sure if people have used this program here, this is anti-smash, which is the one of the programs you can use on the web for analyzing biosynthetic gene clusters. So basically, you enter the genome of Aspergillus calidoostis, and the program will then spit out at you. These are all the putative BGCs in that species. So there are 68 of them. And if they happen to be homologous to known BGCs from other microorganisms, the program will tell you. If not, it will say they are of unknown function. And it turns out that one of these BGCs in this Aspergillus species, shown here, has quite high sequence homology to amidacelamide production in another Aspergillus species. So together, that gives us a very high confidence that indeed this species is producing this particular natural product. We see evidence for it in the mass spectrum, both the molecular ion and fragmentation. And we also see there's a BGC that looks like it should produce amidacelamide. And if you want to, we could then clone this entire gene cluster into some other host species and see if we can then induce amidacelamide production. And this is, in a way, the power of mass spectrometry and analysis right now for natural product identification. We are doing all this without even seeing the natural product. They are present in minute amounts in those samples. You could not practically isolate them even from multi-liter fermentations. But nevertheless, from data analysis, you have a good idea what that compound is. And something more traditional, one of the other peaks that we had identified, and this will be of interest to the students, we found this compound in the other species. And when we tried to match in the database, we couldn't find any natural products that really matched this molecular ion. So then we had to resort to the more traditional methods of doing a large-scale fermentation and isolating the product so that we could do more conventional NMR spectroscopy. And this gives you an idea of the effect of this adding the epigenetic drug. In a normal fermentation, we can only isolate 1 or 2 milligrams. When we add the drug, this is now increased more than 50-fold. And it turns out that when we analyzed it more carefully, and now that we had enough of the material, we could look at the NMR, and the NMR was surprisingly simple. We could only see 8 signals, for example, in the carbon-13 NMR. And the reason is that this is one of those cases where in the mass spectrum, it mainly gives you a dimer. So we were chasing after the wrong molecular weight. And here, under different mass spectral conditions, you can see predominant amount of the monomer, the molecular ion as well. And once we knew that, we could immediately identify this well-known fungal metabolite. So to summarize this first part of the talk, with these epigenetic inductions, you can activate or silence BGCs, and you can do quite high-level analysis using metabolomics, and you can see quite big increases in metabolite production with these epigenetic drugs. And for the next part of talk, I'm going to switch to a quite different organism. This is Eugylema gracilis, which probably everyone here has seen at some point in ponds and other still waters. You can see these green things floating on the surface. They are usually these types of microscopic green algae. And Euglena is quite a fascinating organism. It can grow photosynthetically using just light and carbon dioxide. It can also grow in an animal-like way if given a carbon source. And it's even grown on an industrial scale in Japan as food. And it's even investigated in outer space as a possible food supply for long-term space missions. But from a natural product point of view, it's extremely uninteresting because despite all this work by centuries, there's only one natural product, this terene heterocycle, that's been reported from Euglena gracilis. Nevertheless, we got interested in it because my colleague Rob Field did this transcriptomic analysis of Euglena gracilis. And there is no genome available, by the way. That's because the Euglena gracilis genome is actually much bigger than the human genome. And it's a very complicated organism to sequence. And from the transcriptic analysis, it suggests that Euglena gracilis makes almost doubles the number of proteins of a human. And in this proteome, we also see the signatures of biosynthetic genes like polyketide synthetases and non-ribosomal peptide synthetases. So this suggests that the organism is capable of producing interesting natural products, although none have been reported today. So we grew Euglena gracilis under photosynthetic conditions. So this is like a minimal medium. All you need is some amino acids as a nitrogen source, some inorganic minerals, plus a few vitamins and light, either sunlight or irradiated light, and Euglena gracilis will grow very happily. And for a number of months, Mohammed was doing this, and we were getting more and more frustrated because when we look at the HPLC profiles of Euglena gracilis, we see nothing. Well, we see a few common photosynthetic pigments, but nothing else of interest. And we tried the changing fermentation conditions. We tried adding epigenetic drugs. Nothing was working, and we were getting more and more frustrated, especially Mohammed more than me. And eventually, the only thing that worked was when Mohammed looked into the literature, he noticed that amino acids, when added to Euglena culture, can either inhibit or stimulate growth. And typically when you do fermentations, you don't add amino acids singly, we use complex mixtures. So then Mohammed took out these mixtures and added just single amino acids. And finally, we saw some interesting results. So when you add, here's like the minimal medium, there's practically nothing there. And when you add these amino acids, you can see some of them lead to these new peaks being formed. So this is an example of a metabolite being formed by changing the fermentation conditions in a very specific way. And of course, this will be familiar both to staff and supervisors. This looks nice, but when you do more careful HPLC, you actually see it's composed of about 10 different peaks of different intensities. So then you have to carry out repeated cycles of chromatography and purification to obtain these compounds. But before we got that, again, mass spectrometry gave us a molecular formulae for these compounds. And at least we knew by searching the database that these were novel compounds that had not been seen before. And there was also univisible data that suggested they would contain trilene and perhaps amides, given the number of nitrogens, and also suggested by the carbonyl region as well. So small scale, this was not enough, so we had to do large scale fermentation, 18 liters. That gave us about 200 milligrams of the mixture of those 5 compounds. This had to be repeatedly chromatographed and purified, and that then gave us a few milligrams of each of these compounds. And so finally, we were able to start doing the structure elucidation. And we have a publication currently under revision, but none of this is public. I thought this auspicious conference could be the first time where I could share with you the structures that I'm going to show you in a moment. So the structure elucidation was based, as you probably all would guess, on extensive 1D and 2D NMR experiments. And this is for one of the natural products, the main one. And this is based on COSI and HNPC correlations. So you can see in the growth structure that this is a cyclic peptide. And in fact, the majority contains 5 amino acids, and except for this asparagine, the rest are all unusual non-proteinogenic amino acids. And there's this very unusual 20 carbon long chain fatty acids with a number of chiral centers. So when we saw this structure, we were very excited. Looks very unusual. And afterwards, when we searched the literature, we discovered that there is, in fact, somewhat similar structure, at least in this lipophilic portion. And those are these peptides that were reported from the nematode worm, C. elegans. And this is quite unusual. Those nemamides are, in fact, the only example of a polyketide non-ribosomal peptide mixed metabolite obtained from an animal species. And the Butcher group who reported this identified the biosynthetic gene cluster in C. elegans and also other worms as well. And again, this is a testament to technology. From 50 liters of worms, they isolated 0.07 milligrams of natural product and did all the 1D and 2D NMR and completely solved the structure of their compound. And perhaps because they had so little, they didn't report any bioassays with their molecules. So we tried very hard to get X-ray quality crystals with our natural products, but they were amorphous materials. So then we had to use degradation chemistry to assign the stereochemistry of these different amino acids. And not everyone might be familiar with this. This is a standard method called the Murphy analysis, where you hydrolyze the peptide into amino acids, and these are then reacted with this chiral molecule. And depending whether you have one enantiomer or the other, they will react and displace this fluorine and give you these diastereomers, and these can be separated on the HPLC. And this was first done by Murphy at the Carlsberg Brewery Laboratories, and this is actually a graphic from his original publication. And you can see here, for example, for aspartic acid, this is one diastereomer, and here's the other, separable nicely. And here's the pair from L-phenylalanine and D-phenylalanine. Our natural products contain asparagine, so this will be hydrolyzed to aspartic acid. And then using the conventional Murphy analysis, we can determine the stereochemistry of these asparagine residues, and that's shown here. So this one example has one of them within the skeleton, the other has two side by side, one L and one D. That's the easy part. The rest of the amino acids are all unnatural, so we have to use more complex detection methods for them. Firstly, there's this beta amino isobutyric acid, and this is a beta amino acid, so it doesn't give good separation in Murphy conditions. And in fact, in the literature, there's a paper here recently published where they used this mathematical formula to deconvolute the poorly separated peaks. And we had to do better than this. So Mohammed came up with a better separation method that allowed us to identify the enantiomer present in our sample. For the dihydroxynorvaline stereochemistry, we in fact got a sample from Professor Thierry Gafflot, who made these materials through a bioenzymatic cascade. And using all these methods, we could identify that the remaining stereocenters in these parts of the molecule, that still leaves us the C20 side chain. And that is impossible by Murphy analysis because this just breaks down during hydrolysis. And even if it was not breaking down, there's no suitable reference standard for such complex amino acids. So then we did comparisons with the nemamides and basically concluded that our stereochemistry was exactly the same in that part of the molecule as the worm natural products based on CD spectra and also the HNPC and coupling constants in our molecule. So that's the full structures of our compounds. And then bioactivity, we discovered that these compounds are quite potent antifungal compounds, as well as anti-proliferative against human cancer cell lines. And we also, interestingly, found that we can kill the producing organism, the Euglena gracilis, by adding the compound. We could also add it to C. elegans that produces the nemamides, which have never been investigated in terms of bioactivity because there's not much available. But we can now add our compound to C. elegans, and we also show that it has this inhibitory effect. We can't do BGC analysis for our compounds, unfortunately, because the genome for Euglena is not available. This is what the butcher grip did for nematodes. So the next best thing we can do is molecular network analysis. So this is based on mass spectral data from 3 different Euglena species, and we are looking for related metabolites. And again, if people here are not aware of this resource, this is another great public resource where you can upload your own mass spectral data and analyze it in this way, set up by the Doris Stein group. So this is a molecular network analysis, which tells us that, in fact, these Euglena species are producing about 40 plus natural products in this family. Now, many of them are in too low amounts to practically detect at this moment. So there's only these 5 that we have isolated and purified and characterized. But it suggests that these compounds have some important functions if they are being produced ubiquitously in algae. And meanwhile, there's also a homologue in C. elegans as well. So to summarize, we have found these new natural products, which unusually and astonishingly have this similarity with the only NRPS PKS natural product known from an animal species. And we don't know what that means in terms of evolution or function at the moment. And finally, I'd like to acknowledge not only Mohammed, but all the collaborators. This type of work involves a lot of people in different fields, so I'm grateful that I have colleagues in my university, but also in other countries as well, who have played an instrumental part in this project. And with that, I thank you all for listening, and I'll be very happy to answer any questions.
Thank you very much for a very nice lecture. Actually, I am working on marine microbial, marine actinobacteria as the sponges. And one of the main problem of our group is to produce enough material to separate them. And I saw one paper from High Group, I think in science, that used the same strategy that you mentioned. From the gene map, go to identify the metabolites. It's a little difficult for me to understand, because some compounds are very complex, and for instance, consider terpene, taxol, it has several steps to be produced. So how can we do this job? I mean, LCMS is straightforward, but maybe we cannot detect everything, we cannot identify, we need to separate the compound, then identify the compound. Some compounds are not elucidated, and they cannot be produced. They need a elicitor to be produced, and this stuff. But how can we just identify the second metabolite out of this huge genetic map? This is one of my questions. The second is that what is the basis of the selection of the elicitor? Because the plant, for instance, when they are attacked by herbivore or macro, there are 2 different pathways of elicitation, nuclear adjustment, and so on. What's the bacterial? Just elicitation methods. Thank you very much.
Thank you very much. Those are both complicated questions, but I'll try to answer briefly. When it comes to elicitors, there are many different elicitors you can use. One are the epigenetic molecules I showed. but there are literally thousands. And there are groups that have done experiments where you grow the microorganism plus an elicitor and try like 10,000 different elicitors in parallel. If you have the processing power to process each of these fermentations and look at their metabolite profile, that can be a very powerful approach. And if you are interested in that, I would recommend the very nice review recently by Mohammed Sayed Dost in Princeton, which covers this elicitor approach in much greater detail. It works. The problem is you won't know which elicitor is going to be the right one for your strain. So you have to try lots of them, right? And usually something will work, but it's an empirical method. The other question is regarding these BGCs. So if you have a genome, and this is the other thing that's also perhaps worth mentioning, that genome sequencing has become very practical and cheap nowadays. So if you can grow your actinobacteria, you can get the complete genome sequence for maybe $100, $200. You know, there are organizations that will do it for you. And once you have the genome, you can put it into the software program like anti-smash, and it will tell you what all the putative biosynthetic gene clusters are, based on the types of enzymes present in those clusters. And what it will tell you, and I'm sorry that if I'd known you'd ask this question, I would have given some better examples in the slides, but if I go to the one from the Butcher group, so this is their homology analysis of the PKS1, and it's probably too hard for you to see there, but what the program will do is assign function to individual genes, like there's a ketosynthase, there'll be a condensation domain, there'll be a reductase, and so on. So you kind of know what chemistry each of those genes is doing. If you're very lucky and it is very straightforward, you can even predict the structure. Sometimes you can do that, but usually not, because you don't know what your feedstock is. You don't necessarily know what the starting material is that is going into these modules and what will be coming out. And then if you want to know the structure, you then need to be able to detect the natural product. And that can be a major challenge, right? So you need to grow your organism and see what you can find in the mass spectrum and identify structure. You still need to do that yourself, you know? So the gene cluster may tell you there's a diterpenoid present, but it won't tell you that it is taxol. That still needs the chemical analysis, either through detailed mass spectrometry or more usually by producing enough of the compound that you can isolate, purify, and do the normal structure elucidation. So that part is still very important. You can't do without it. But what the mass spectrometry can help you with is guide you towards which extracts or fractions contain something interesting which might be worth further attention. And what they are very good at is also telling you if you can match with the literature. So it's very useful for dereplication. So if it can tell you if this particular peak that you're seeing is the same natural product that somebody else has isolated. So it's good for that. But to confidently assign structure, if it's a new structure, you still need to do NMR and other techniques, which means isolating enough of them. And the problem, as I said, is that usually these BGCs are not expressed. So your actinobacter, if you sequence it, you may find lots of interesting BGCs, but under laboratory conditions, they will not be produced at sufficient amount to be isolated. And that's when you have to resort to these indirect methods. One possibility is to add an elicitor and pray for the best. Another possibility is to take out the gene cluster you're interested in and express it in another host, which can be done with actinobacteria and also with fungi. But each of those experiments requires, expertise in recombinant DNA. And for one BGC, that's probably like 6 months work to get it into another host, expressed, and then you still need to isolate that compound and identify what it is. So these are not things that are straightforward, but they can be done now. And as I'm saying, in the examples I showed, even any simple microorganism, you don't have to look for something exotic. Any microorganism out there will contain 50, 100 BGCs. And most of them, we have no idea what they are producing. So you don't have to look for an exotic microorganism. The real challenge is to take a microorganism and be able to investigate its full biosynthetic potential. And at least now we are starting to make progress. With these types of experiments would not have been possible without genomic sequencing and data analysis and bioinformatics to compare the genomes between species. I hope that helps answer the question.
If there are no more questions, then we can conclude this inquiry and advance talk, multidisciplinary, because we cannot work with isolation alone. We need genetics as well, and this goes very nicely. So thank you very much for your lecture.
And I'll just make one comment. I don't have an enlightened science minister like Professor Ataur Rahman. So those mass spectral analysis we do, it's in our university facility. It costs us $100 US per hour, okay, which is a lot of money. But without that, we cannot do this type of research.
And now I invite you for lunch, and we will proceed at 2 o'clock in the same room here.
Ladies and gentlemen, may I have your attention, please? There is an announcement from organizers that gala dinner is arranged for registered participants tomorrow. और अगर किसी मेहमान ने अपने साथ किसी मेहमान को लाना है तो राही से राब्ता करें।